the day today. Okay, so it is my super big honor to get to introduce Brendan Hobart here giving his exit talk for his PhD. Here's some cute pictures for us. So Brendan hails from the great state of Wisconsin, where he completed his undergraduate and his master's degrees, both at the University of Wisconsin-Madison. He did a pretty spectacular master's degree on the California spotted owl in the Sierra Nevada system, and has a whole suite of work that he's published on that, one of which won a student award from the Wildlife Society as a best paper. During his time here at CU in eBio, he's won an eBio fellowship. He's won an NSF intern fellowship and got to collaborate with the USGS. He's gotten an Excellence in Teaching award. And throughout his time in master's and PhD, so far, he's published 9 papers, which is really impressive in terms of productivity as a graduate student. Brendan is a very accomplished field biologist, as you can see. And here it looks pretty amphibian rich, which is true, but it's also true that he's done tons of work on birds. He's an excellent naturalist, incredible birder, amphibian lover, and he has spent many hours immersed in ponds, as we will see and hear about throughout his talk today. And like a lot of graduate students in recent years, Brendan was pretty impacted and interrupted by the global pandemic that happened. A lot of people have challenging things come up during graduate school, but geez, an entire global shutdown. I'm speaking to all of you who know this. It's pretty challenging. But it's been pretty incredible to watch Brendan's resilience, because the pandemic, as we know, was a lot of isolation. I actually have no pictures of Brendan with other people, just with amphibians. Like, this is literally it. Like, Brendan spent a lot of time alone with amphibians. But he demonstrated so much grit and resilience in coming through some challenging times through the pandemic. And he emerged to become this really incredible leader in analytical approaches and modeling and quantitative statistics, etc. Really hard stuff, actually really hard stuff. So much so that he's now become a helpful mentor to many of his fellow peer graduate students. And so that's been pretty awesome. He's also an incredibly gifted graphic designer. And so when you look at his talk and you see all the beautiful images and figures, he made all those and he designed them all. It's just a natural talent that is really evident. He's really demonstrated artistry in science that is pretty cool. So while the pictures don't indicate it, Brendan is actually a very engaged community member and has been a great person doing a lot of service in our department. He's led the colloquium committee. As I mentioned, he's helping mentor a lot of other students, and he always does all of this engagement with a lot of joy and professionalism. And I personally thank him a lot for teaching me so many things during his PhD, things I didn't know about as much before, like causal inference and Bayesian modeling and birds. Really good stuff. Anywho, did I get the Wisconsin?
Yeah, right.
When you doing it? I thought I didn't. Yeah. Anywho, another thing that's really special about Brendan is his proclivity to ride in style. When he goes to the field, his mode of transportation is this very large camper van, which really comfortable, really all the amenities. But when he goes to a conference, he prefers to roll up in like a hot red muscle car. Literally, this is what he picked me up in Nashville to go to a conference in the fall. Very funny. So he'll be riding off into the sunset at a postdoc back at the home grounds in the University of Wisconsin-Madison. So when you're really awesome at something, people recruit you back and they work hard to recruit you back, which is what happened in Brendan's case. So, all right, without further ado, here's Brendan to share his work.
All right. Thank you, Val. That was far too kind. So, and thank you all for being here. really good to see some friendly faces, some new faces. This is gonna be a bit of a wild ride. Snails to sagebrush is like, those are different. Those aren't the same. Subtitle: Leveraging Field Data to Better Understand the Causes and Consequences of Infection and Invasion in Diverse Systems. It doesn't make it any more clear. And so I want to start with this question of like, why are we here? Some of you are here because you're family and you have to be here. Some of you are here because you're first year graduate students and you have to be here. But more broadly, the operative word here is why. I have always been interested in knowing why things work the way they work in all arenas of my life. But I've also at the same time found a lot of joy and comfort in natural ecosystems. And so it's been a pretty comfortable career path for me to be an ecologist and try to understand why ecosystems work the way they work. And in particular, I've been interested for the better part of a decade now in trying to understand why particular species within ecosystems are declining or disappearing. And as ecologists, when we start to try to answer this question, we start trying to understand how that species interacts with its environment, with its habitats, and with other species in the system, here by drawing in these modular interactions. And then we start drawing in even more modular interactions for how those other species interact with their habitats and with, even more diverse species in a system. And so it becomes a really reticulate, complicated picture. And I argue, I believe that to be an effective applied ecologist, we have to be able to transcend systems and scales and ways of thinking and not get pigeonholed into any one. And I've used my dissertation to try to do that, to span boundaries and to try to understand a lot of different facets of ecosystems. And I think that will hopefully come through by the end of the talk. And then we have to go even further and remember that this single fake frog in this single fake pond is just probably one pond of many within a landscape. When we move up in scale, we have to begin adopting new ways of thinking, new tools, thinking about metapopulations, meta-communities, for example. Just a whole new way of thinking about the world. And then this landscape of ponds, again, is probably just one of many across a broader region or a continent where we need, again, new skills, new tools, new ways of thinking. For example, bringing in concepts from biogeography. And my dissertation, again, very broadly, arcs those scales. That's one of two animations in the whole thing. So if that's your jam, relish it. There's only one more. But I don't work on pretend frogs and pretend ponds. I work on snails to sagebrush. So we're going to cover some really diverse and I think interesting systems throughout this talk. And the first system that we're going to talk about is freshwater snails and their infection and a symbiotic consumer that lives on them. And we're going to breeze through this one because it's been published for a couple of years in the Journal of Animal Ecology with some very fine folks in the Johnson lab, some of whom are here today. So if you want to know more, there's a paper with all of the nitty-gritty details, and I'm happy to talk about it more later. So as Val alluded to, I weathered COVID during this PhD. You also weathered COVID. Congratulations. And so I don't think that I need to tell anyone how important pathogens are or the transmission of pathogens among hosts. And so I didn't study COVID, but I did study macroparasites that infect freshwater mollusks. And for a long time, people have talked about, both with this system or COVID or many others, how the abiotic environment affects the transmission of infectious stages from one host to another. So with COVID, we thought about things like air circulation, humidity, so on and so forth. More recently, and by more recently, I mean in the last decade, folks started thinking a lot more about how the biotic environment affected transmission. This is really cool because it's starting to think about parasites really as interacting members of ecological communities. And I became, in the early days of my dissertation, very interested in how predation or the direct consumption of, it's number 2, of infectious stages could alter host risk or the ecology of those consumers. And I was thinking about this in a system of trematodes. So these are flukes or flatworms are more common names. And they have these complex multi-host life cycles. This is just an example on the left, Riboroia ondatrae. So it's moving from snails to amphibians to perhaps birds back into snails. They cause these crazy limb malformations that I unfortunately won't have any time to talk about today in amphibians, because the part of this life cycle that my work focused on in this chapter was the movement of infectious stages into snails and out of snails. And at this stage, trematode infectious stages are encountering this guy. This is Chetogaster limnaei. It's a worm, a true worm, and a ligochaete. So it's blown up in the bottom left there. And you can see dozens, perhaps hundreds of this worm on the face and mantle of this snail. And so when we were dissecting tens of thousands of snails in the field, we'd see these guys a lot, but it was kind of unclear what their role was in this system. And it was similarly unclear in other systems. Dissecting snails is a rite of passage for trematode parasitologists, and many people for many decades saw these and would make some anecdotal observations, but it wasn't particularly clear what their functional role was. And so we can walk through the life history of this whole system and think about it, and then I'll show you just a blip of results. So trematodes can infect snails in one of two ways that varies by taxa. They can infect actively with these swimming stages, or passively with eggs, where the snail consumes the eggs hatch inside the snail, and they're upon infected. When we add Chetogaster into this picture, we know that Chetogaster eats these active stages, and that this can yield protective predation of the snails. That's what we coined it, at least. So having this consumer around, living all over your face, kind of gnarly, kind of gross, but it's protecting you from infections. On the other hand, these passive infections do still occur. When they do, they get to the snail's gonads, they undergo several rounds of asexual reproduction, and then they begin releasing this next infectious stage. Again, this would be going to find the next host, for example, a larval amphibian. And once again, we know that Chetogaster eat these outgoing infectious stages, but these infectious stages are super abundant in many cases, and they're very nutritious. And so, so we know that Chetogaster increases in population size when these infectious stages are being released from snails, and we coined this predator augmentation. And again, it was unclear whether we would see protective predation or predator augmentation in the field in a big data set of observational data. And what was really neat in this study is we saw both, and it depended on the taxa of trematode, depending on whether it infected actively or passively. For the active infectors on the left, we saw a signature of protective predation. And for the passive infectors via eggs on the right, we saw a signature of predator augmentation. And this was the first study that had found both of these results at once by leveraging this information about life history of trematode infection style, infection mode. And so this work was not particularly applied. Mostly it's just a really fascinating ecological system to study and make questions in, but there are, some potential biocontrol applications of Chetogaster, or at least that has been suggested. So schistosomiasis is a trematode that is a major health concern, largely throughout Africa, that infects actively. And so there's a chance that trying to increase Chetogaster populations could help control that pathogen. And then more close to home, swimmer's itch, more formally cercarial dermitis, is trematodes burrowing into your legs or your exposed skin. So sorry if you didn't know, see some faces. But maybe if there's more Chetogaster around, we get less swimmer's itch. That requires a lot more work, but it's really interesting avenues. And the Journal of Animal Ecology thought it was interesting as well. They covered this work in a research highlight that was written up by Janet Kaprivnikar, very talented writer. And so this highlight might be even more digestible than the original paper that we wrote. So you have not just one, but two places to go read more about this if you're interested. Okay. I told you it'd be a wild ride. Hold on to your butts. We're gonna leap now. We're gonna keep the host pathogen theme the same, but we're gonna leap up in scale, and we're gonna think about a different host pathogen system. In the second chapter, which is titled Hosts, Pathogens, and Hot Ponds, Thermal Variability and Heat Contribute to Spatial Patterns of Chytrid Infection. And I just want to thank my co-authors up here, many of whom are at the USGS in Madison, Tim Porpita, former member of Val's lab. Val is on this project despite her photo not being up here. Sorry, Val. I meant to do it right before the talk and then. Classic. This one's not out yet, so I'll cover it in a bit more detail. So as many people in the room know very well, as climate change progresses, we expect to see increases in mean temperature and temperature extremity and variability. And all of these shifts can affect host pathogen interactions. For example, by shifting the ranges of hosts or their vectors leading to novel host pathogen interactions, but also by changing existing interactions, by changing the playing field within which pathogens and parasites are infecting hosts. And a model system for understanding these sorts of shifts and impacts has been Batrachochytrium dendrobatidis, or BD, as I'll call it for the rest of this portion. This is a fungal pathogen that infects amphibian skin. It is a conservation concern around the globe. It occurs on every continent that amphibians are found on, and it's caused declines and extinctions in at least 500 species of amphibian. And so over the last couple of decades, a lot of work has gone into understanding where this pathogen is and what it's doing. And interestingly, there's a lot of variation in which species it infects, how much it infects them, and what sort of pathology results from that interaction. And from the start, temperature has been suggested as an important driver of such variability across space and across time. And in very broad brushstrokes, there are two predominant patterns that are seen in the literature and across study systems. And the first is that as conditions go from more stable to more variable in terms of temperature, we tend to see more BD on more hosts. The reason for this is physiological. Pathogens adapt and acclimate to new temperatures faster than their hosts. And so when we have wildly fluctuating temperatures, it favors pathogens, at least for some period of time. The second main pattern is that as things get hotter, we actually expect to see less BD on fewer hosts. And again, this is grounded in physiology, but more in terms of the upper thermal limits of species. And taxa, BD, like many fungal pathogens, is relatively heat intolerant, and especially compared to most of its amphibian hosts. And so when things get hotter, amphibians tend to persist, but BD does quite poorly. Most of the work that's gone on at these, or to inform those two patterns, has occurred at really large spatial scales, so thinking across continents or regions, or really small spatial scales, thinking about individuals. Was this frog caught in the sun or in the shade, and does it have BD? There has been comparatively less work at the landscape scale. And just fundamentally, this is an interesting scale to work at as an ecologist. It's a fertile ground for discovery, but it's also really important for management. This is the scale at which managers are typically, or very often, making decisions. They have some number of ponds or other breeding sites, and they need to make decisions about where to prioritize management or conservation or monitoring. And so it's at this scale that we set out to answer two questions. The first is very simple, and we wanted to know, when we're working at the landscape scale, looking at multiple host populations, do we see relationships in the directions that we would predict between variability in heat and BD? And then second, if we do see relationships, what is the important source of variation in temperature for driving those relationships? Is it seasonal changes in temperature that emerge as a relationship with BD, or is it differences across space, differences across sites within a landscape that drive relationships? And this is, again, also relevant to management, because if it's the latter, if it's spatial differences, that variation in temperature can then be leveraged for management. And so we set out to answer these questions in a system of 23 ponds in southeastern Wisconsin. You can see based on the scale bar at the bottom that they're all quite close together. They share a climate, and they share pretty similar land cover characteristics, but with subtle differences and certainly differences in temperature. They also have a really cool, cute amphibian breeding community. So you can see some representatives of different genera along the bottom. The species, though, that we focus on is eastern newts, Notothalmus viridescens. They're sort of the common denominator in terms of aquatic amphibians in these ponds. And I'll note here that BD is not highly pathogenic, which for this species in this system, which does change kind of how we think about and interpret results. So we sampled those ponds and those amphibian species in 2022 and 2023. Before the breeding season, we would go and we put out a temperature logger that was taking hourly readings. And then we would perform anywhere from 2 to 9 follow-up visits to each pond. At each visit, we would net amphibians to capture them to get density estimates, collect water data, and then importantly, we would swab amphibians. So much like we got our noses swabbed for COVID, remember that BD is a skin pathogen of amphibians. And so when we swab its skin, we end up with some BD DNA on those swabs. We can process those swabs using DNA extraction followed by qPCR to understand at the individual level whether newts were infected or not and how much BD they were infected with. And we paired those infection data principally with temperature data. So these are some loggers and there's a PVC tube that they were deployed within. And recall that we wanted to capture and try to understand both heat and variability. So we made a bunch of different metrics to think about each one of them. We explored those in different rolling windows. We used myriad tools to understand which were related and which captured these processes at best. And what we landed on were, I think, 2 kind of elegant, simple metrics. For heat, we took the sum of hourly temperatures in the 8 days preceding a sampling event. So this is capturing thermal input into a pond. And then for variability, we took the standard deviation of our hourly temps in the 8 days preceding a sampling event. So how bouncy is temperature leading up to an event where we swab a newt. What I came to realize, perhaps not quickly enough, is that these 2 variables are complexly related to one another. Where more variable ponds are also the hotter ponds and vice versa. So just to illustrate this for you, this is a snapshot of all 23 ponds temperature through a couple of weeks in both years. Each pond has its own line moving through time. I'm going to highlight now the most variable pond in red. And if you squint, you can see that it also tends to be the hottest pond. We can see this even more clearly if we look at a scatter plot of these 2 variables against one another, faceted out by time. And so I needed a way to encapsulate both of these variables together, because the reality was that in this system, you can't separate the contribution of these 2 variables to BD outcomes. They're just operating together. And so I came up with this index to roll them into a single variable where we z-scaled each one because they have quite different ranges of values in their native format. And then we took the mean of those 2. And so this creates a surface where as we move from yellow to purple, we go from cool and stable to hot and variable conditions. We can map those index values back onto all of our pond visits, all of our sampling events. And then we can incorporate those values in a generalized linear mixed effects model. I won't cover this in great detail, but individual level BD load, again, from swabs is the response that we're explaining with a bunch of different covariates. We have controls for spatial confounding, temporal confounding, spatio-temporal confounding. We're counting for host density. And then importantly, we have the index and its square to allow for a quadratic and nonlinear relationship between temperature conditions and BD load. So again, if anyone's interested in modeling for this chapter, for the first one, for the one that comes up next, just chat with me, because I'm going to skim over most of the methodological details. So to orient you to the important results here on the y-axis, we're going to have the predicted BD load. So that's how much infection does an individual newt have. And on the x-axis, we have our index, where we're moving from cool and stable to hot and variable as we go left to right. And what we found was this really clear, but also confusing, nonlinear relationship between these variables. And I think that we can use the 2 underlying metrics of temperature characteristics to explain this, where I think it's most plausible that variability is driving the left side of the curve. Again, there's a lot of evidence suggesting that as conditions become more variable, we would expect to see more BD on hosts. But recall that as we make things more variable, we also make things hotter. And so I think that on the right side of this curve, heat drives a downward relationship where we reach some threshold, some point where conditions begin exceeding BD's upper thermal limits. and it starts doing more poorly on hosts, and we see this tailing off. And so the answer is a little complicated and a little, it leaves something to be desired, right? But this is field ecology, these are field data. It's always going to be challenging to dig in and attribute patterns to mechanisms. But it does let us move on to the second question, which again is trying to understand what the important source of temperature differences are. Is it temporal or is it spatial? We know that we have a pattern between BD load and temperature, but where is the important and relevant temperature variation? So the original index that I've already explained allowed thermal measurements to vary both temporally and spatially, right? But we can make alternative indices that isolate those respective sources of variation. We can make one that's pond specific and isolates temporal variation. This is isolating seasonal changes within a given pond. So now a high index value is hot and variable relative to all other time points at a given pond. We've removed the spatial component of temperature. Similarly, we can make a time-specific index that is now isolating spatial differences. So now here, a high index value means hot and variable relative to all other ponds at this given point in time. And so we made these alternative indices isolating temporal and spatial differences in temperature conditions, respectively. And we fit those same models with them. And we asked which one of these could recover the original relationship with BD load. We did some number things to validate this, but just visually, I think we can all see that the index isolating spatial variation is the one that replicates and recovers the relationship with BD load that we saw when we allowed temperature variation to be both spatial and temporal. And in this system, the effect of temperature on this host pathogen interaction is not so great that we end up with asynchronous ponds or ponds that remain infection-free throughout the season. No, instead, all ponds follow this similar trajectory of BD load through time. These are real observed means per pond. But it is driving temperature differences among ponds are driving order of magnitude. These are order of magnitude differences between ponds in their mean host pathogen outcome. And so this is really relevant, again, I would argue, to management. It's been suggested in multiple systems that we could directly manipulate the thermal environment to change amphibian BD interactions. For example, by clearing trees around a pond, letting more sunlight in, warming things up so that we hopefully reach the right side of that curve and see a tailing off in the amount of BD infection per host. More generally, this has been really cool research in bringing together concepts, again, from both small and large spatial scales to tell a story at this landscape scale. And leads nicely in this, again, increasing arc of greater and greater scale to the third chapter that I'm going to talk about. This is a shift both in scale. We're going to begin thinking across a large region, and a shift in system and in taxonomy as we're going to be thinking about birds and bird communities. So this chapter is titled Annual Grass Invasion is Transforming the Sagebrush Biome's Songbird Communities. And again, I'd like to thank a cool, diverse team of collaborators and co-authors on this project. Again, a couple of whom are here. So yeah, thanks for turning out. Thanks for making this project much cooler than it otherwise would have been. So the sagebrush biome it's vast, covers most of the Intermountain West, much of the American West, in fact. Habitat here has been lost to the normal culprits, energy development, housing development, agricultural development, over the last century or so. But probably the most pressing, the most dire, the most challenging contemporary conservation issue is the invasion and dominance of non-native annual grasses like cheatgrass. And we can see just in the last few decades, between 1988, 2018, that these species, again, predominantly cheatgrass, are really increasing their dominance across this region. We're just seeing more oranges and reds where these species are becoming abundant. And the outcome of these invasions is really quite sad and really quite dire and drastic. So this on the left is an intact sagebrush habitat. It's dominated by sagebrush shrubs. There's some other shrubby woody species, and then there's some perennial grasses in the interstitial spaces between those shrubs. It's relatively diverse for a dryland system. But again, cheatgrass, which is this yellow golden grass, is invading these systems. It begins by filling interstitial spaces between all of the shrubs. It begins competing with native plants for resources, primarily for water. These are water limited systems. And it changes fire regimes pretty dramatically, where we see far more frequent fires within invaded areas relative to uninvaded areas. And whether it's through competition or fire, a combination of the two, the outcome can be and often is a near monoculture of cheatgrass. And it's, I think, just visually striking how simplified these systems can be and how far departed from historical conditions these systems can be. And this sort of dramatic change is something that ecologists have noted for a long time. We would be doing a bad job if we hadn't. And they've been that sort of change has been given many names over the years, but recently discussion of these changes and how to manage them has been crystallized under the concept of ecological transformation. These are some classic or highly talked about ecological transformation examples. On the left, we have a ponderosa pine forest becoming dense as we suppress fires, burning to the ground, and then seemingly permanently transitioning to a shrubland. This is happening all throughout the West and Southwest US. On the right, we have native prairie, the left side of this dashed line, that has been encroached upon by woody species in the absence largely of fires and changes to fire and disturbance regimes. And so, as is captured by these two examples, most of the work to date in the last 5 years or so on ecological transformation has focused on plant communities. This is pretty natural. They're highly visible, viewed as foundational to ecosystems, and importantly, we can measure them and their change relatively easily using remote sense data at really vast spatial scales, because ecological transformation, it happens locally, but it's a regional and often continental problem. By comparison, animals have not really entered the discussion about ecological transformation. And that comes down to a number of logistical challenges. Again, it's much harder to measure animal communities at scales relevant to transformation than it is to measure plant communities at those scales. And that's not to say that people haven't thought about it. Of course, wildlife biologists have thought about how sweeping changes in vegetation communities have affected wildlife. But wildlife and animals have not entered this recent discussion, right? And that's important for a couple of reasons. The first is that these communities and species and populations are almost certainly affected by transformation, and they probably affect transformation themselves. Animals have a suite of diverse and important functions that they play within ecosystems, right? Think about seed dispersal, pollination, herbivory, predation. And if we change animal communities and subsequently change that functioning, it could have cascading consequences back onto habitat change and vegetation change. Those changes or those feedbacks could be dampening or amplifying, but we really need to start from the perspective of how are animals responding to and being affected by these sweeping ecological transformations. And the closest thing that we have in the sagebrush system is sage grouse, which you can see here. Sage grouse are something of a hot button topic in this region. They absorb much of the conservation focus and funding and management. And for good reason, they've declined dramatically since European colonization of the sagebrush biome. And they're viewed and talked about as an umbrella species, a flagship species, implying that if you protect them, you're protecting a host of other species that have similar niches. In some systems, that's a pretty okay implication, a pretty fine assumption. In this system, it's only okay. We know from an abundance of empirical research that Sage grouse are a modest at best umbrella for other species in the system, even for other birds using sagebrush habitats. And there's a diversity of such species. This is just a snapshot of representative songbirds that are home in the sagebrush biome. And unfortunately, we don't know how these species generally are responding at a regional scale to annual grass invasion. That remains a big question mark. And that's too bad, both from the management and the conservation of these species. And we can't really hope to understand their functional contributions and how those contributions will change if we don't first just understand how they're being affected. And so this chapter focused on understanding how grass invasion is affecting 40 songbird species that breed in the sagebrush biome. We selected 40 species that routinely nest or forage within sagebrush habitats. And songbirds were a nice choice here as opposed to raptors or other taxa because their home ranges are small enough to adequately be covered by systematic point counts. The systematic point count survey data that we used, I didn't collect this myself in the last year, was from Bird Conservancy of the Rockies. They've been surveying this hatched region for about a decade, really providing a great service to the community. And we paired those exhaustive and systematic survey data with remote sense data that captured everything from human impacts to climate conditions and land cover. And the first thing that we tried to tackle when pairing these two sources of data, which are observational, was confounding, right? So again, we are broadly interested in the effect of invasion on birds. But we know that invasion occurs non-randomly. For example, there are strong elevational patterns to where annual grass invasion occurs. And we know that elevation independently affects bird communities. So this is classic confounding. If we don't control for the effect of elevation here, we will obtain a biased estimate of invasion on birds. And so one of the tools that we used to address this confounding was pre-regression or pre-analysis matching. And just to sort of provide a really graspable example of this, consider this toy landscape that increases in elevation as we move left to right. Each one of these squares is a survey site. So somebody's gone out and counted birds at each one of these. And you can see a rough elevational pattern here where we have more light colored sites. These are uninvaded at high elevations, and more dark colored sites that are invaded at low elevations. And really simply, heuristically, what matching does is identifies pairs of sites that differ in their invasion status, but are similar in terms of their elevation. And then we discard the extreme sites that don't have an analog. There's no good comparison at those sites. In reality, we do something far more complicated that allows us to match on a bunch of covariates and not just elevation. These are all things that we thought were plausible confounders affecting both the distribution of annual grasses and bird populations and bird communities independently. And this matching this is called propensity score matching. Again, this is a detail that I'm going to glaze over, but talk to me or Laura if you want details. Probably talk to me. So this matching was super successful, and there's a lot of information being shown here. But the real point is that the red points, which is the difference in these underlying covariates between invaded and uninvaded sites before matching, these sites, these populations of sites were very different from one another. Those red points are far from zero. But after matching, the black points, we shrink all those differences. And so what we're doing here is we're making our set of sites that are highly invaded, our set of sites that are uninvaded or lowly invaded, we're making them much more similar to one another in terms of all of these underlying conditions. And we can visualize this because the plot on the left only goes so far, but these are distributions on the top of summer vapor pressure deficit before and after matching at uninvaded and invaded sites. And we can see that before matching, there were many more high VPD, so dry sites that were invaded, but we shrink the difference between those two different distributions after matching. And the same thing for elevation, where again, we had more uninvaded sites at high elevation before matching, but we shrink these differences by excluding extreme sites by finding pairs of sites that are similar to one another in all factors except for invasion. And what we ended up with after this exercise was about 500 survey sites that had matches. Those survey sites are shown in blue squares, and they came from the population of smaller black points here. So covering, again, a lot of the sagebrush biome. And at each one of these sites, we used a model that at each one of these sites estimated very many parameters. It was a Bayesian multi-species, multi-scale occupancy model. This is probably the biggest disservice to my methods that I'll do because I'm only going to tell you about 2 of the parameters that model estimates to keep things simple. The first parameter of relevance is psi. This is a pitchfork looking symbol. And this is estimating or capturing occurrence of species. So it's asking, does a species occur in one of these 1 by 1 kilometer squares or not? So it's capturing a species broad scale distribution across this, very, very large region. The second parameter of interest is theta, the circle with the dash through it. And that is asking, when a site is occupied, what proportion of the points within that site have the species? To put it a little less formally, it's a species prevalence, and it's working towards a species pseudoabundance. How common is it where it occurs? Species could occur somewhere and yet still be rare, and that would be low theta. And what's cool is that this model estimated these 2 parameters not only as a function of covariates like annual grass cover, but for all 40 species in the study simultaneously. And so that produced an insane amount of results, a crazy amount of data. And what we did to simplify this and coarsen things a little was group the 40 species into 6 nesting-based guilds, nesting habitat-based guilds. So we have grassland, woodland, generalist, riparian, shrubland, sagebrush. These are all mutually exclusive. And for each one of these groups, we could think about the average response or the average effect of annual grasses on psi and on theta. And we can think about community structure. What does an uninvaded community look like in terms of its contribution of all of these guilds to the community versus an invaded site? And so we can get into those 2 sets of results. The first, again, is just asking, what's the effect of annual grasses? The label is annual foreign grass. Don't worry about that. It's annual grasses. So the first is asking, what's the effect of annual grasses on these species occurrence, solid lines, and prevalence, dash lines. For grassland species, we can see, it's a little hard to see, but there's a positive effect of annual grasses on these species occurrence. We can fill this in for all of the guilds, and even though we've coarsened this, we still get a lot of information. And one of the big patterns here is that the 3 guilds on the top, their occurrence, psi, the solid lines, is positively affected by annual grasses. So these species ranges are actually broader within invaded areas. On the bottom, though, we see these 3 guilds are negatively affected in terms of their theta, their prevalence by annual grasses. And I want to highlight this bottom right panel especially. These 4 sagebrush species are endemic to this region. They occur nowhere else on the planet. They are important conservation targets. And this pattern that we see here for sagebrush and shrubland and riparian species, this pattern of a decline in theta with relative stability in terms of psi, points to an ongoing shift that's occurring for these species populations. In the face of invasion, we would expect initially an erosion of species abundance, right, within these areas. We would just have fewer individuals. They would still occupy the same broad area, but we'll have fewer and fewer individuals, and that's what this is signaling. And eventually, if this continues unabated, it would probably lead to declines in occurrence as abundance reaches too low of levels for these species to persist within landscapes. And we can put those differential responses together to ask again how community composition now varies as a function of annual grass cover. And what we see is I think quite striking. We see as annual grass cover increases, increasing dominance by especially grassland species, and to some degree, woodland species and generalist species, with concurrent declines, especially in shrubland and sagebrush species. And so this is pointing to a concurrent transformation of sagebrush songbird communities alongside sagebrush habitats. But remember that the broader transformation is indeed broader. It's happening across an entire biome, entire region. And so we wanted to make some inference at that scale as well. We started this by gathering relevant covariate data across this entire region. And we used these covariate data to make some predictions. And the prediction that I'll focus on here is on the left. This is predicting the diversity of species, bird species, that are negatively affected by invasion. So we went into our model results. We said which of these species are net negatively impacted by invasion, because these are species that we probably need to think about, right, as invasion worsens and expands. And we wanted to take it a step further by asking, where is invasion actually likely? And so we gathered an independent source of ecosystem resistance to invasion. And so high resistance in blue, these are areas that are relatively unlikely to become invaded in the future. Low resistance areas in red are relatively likely to become invaded, and green is intermediate. We can spatially intersect these two raster images and obtain this third panel, which is diversity parsed by resistance class. And this exercise has been, I think, really important, and this sort of exercise is relevant to management, because the optimal management probably almost certainly differs between these different predicted areas. So consider dark blue. This is an area with a lot of species that are negatively affected by invasion, but where invasion is pretty unlikely to occur. And so these are probably areas where it's cost effective to try to resist invasion through early monitoring efforts, through spraying of any propagules. And that differs dramatically from dark red. These are areas with a lot of species that are negatively affected by invasion, but where invasion is probably going to happen whether we want it to or not. And so these might be areas within which we can direct that invasion and that transformation. For example, planting native species that we know are important to birds, suppressing fires that remove really dramatically and rapidly the shrubby component from these ecosystems. And so this has been an important exercise. I think this paper is going to be really relevant to kicking off thinking about animals in this transformation, and hopefully in transformations more broadly, because we've shown that as habitats, as vegetation communities transform, so are animal communities. The next step in this system and in many others is going to be thinking about the functional contribution of animals. It's likely that the birds that we are losing from these ecosystems provide important functions that could stabilize these plant communities. And if we lose these birds, we probably have a greater chance of losing even more vegetation, native vegetation. So that was the wild world of my dissertation. I promised you it would be. I'm going to close just by saying that it's been a lot of fun to work across these diverse systems. In each one, I have learned a lot about new species. I have met new people, new collaborators. I've got to travel to cool, often muddy field sites. Val demonstrated that very nicely. And it's, I think, been very effective in making me a well-rounded applied ecologist, but perhaps you can be a better judge of that. We've lost clicker. Nope, don't do this now. All right. And I also think, as I asserted at the start, that working across broad scales like this has been super valuable in shaping how I think about ecosystems, being able to zoom in and zoom out and try to understand which information needs to be retained on each one of those zooms is an essential skill that I think will probably serve me well moving forward. And it's kept me from getting bored. No chance of getting bored in the last 5 years. And more broadly, I hope that the research that I do, and I think the research, other research like that, which I do, is relevant to trying to understand how to effectively manage animal communities within transforming ecosystems. So our pretend pond is now eutrophic and somehow on fire. But animals persist. Animals always persist. Not always. There are some notable examples, but animals often persist. And I think that research like this can help us understand how, and again, why, and lead us towards more effective management. So I have a lot of people to thank. I pride myself on low text slides. This isn't one of them. And I'm not going to read all these names, sorry, but it's just to say and demonstrate that there's a big community of people here in eBio that have been nothing short of incredibly supportive in the last 5 years. Chief among them in recent years is Val. So thank you very much. This is, I thought this was the only photo of us, but there's actually the one with the charger, the muscle car. So there's two photos of us, 2 animations in the whole thing. There's also a bunch of non-eBio people, both within and outside of CU, who have contributed to my science and professional development throughout this time. Many were co-authors on the projects I just talked about. I also had a number of incredible field crews throughout the years who aren't co-authors often on these projects, but really helped me keep my stuff together out there. I want to thank my family, many of whom are here today. They've weathered a lot of conversations of parasites over dinner, which I think is great. They don't think so as much. Yeah, so thank you all. Bunch of friends from back home in Wisconsin, some of whom have moved out west and even further west, have helped me get up to some good stuff throughout these years and served as a reality check that there's more to life than dissertations. And the same goes for Colorado folks, some of whom are here today, who helped me get up to no good. Yeah, kind of like constantly no good in a lot of different ways. So thank you guys. And then finally, Aaron has been like at the very center of my support network. Yeah, we got married in the middle of the first Wisconsin field season. Like I was doing sampling the week of our wedding. So thank you for letting me do that. It felt right. I wouldn't suggest planning a field season and a wedding, but we pulled it off, and then my bird friends as well who have to make it in here. Yeah, so thank you all for being here. This has been a ton of fun. I think we have some time for questions. Yeah, so I'd love to take some questions. Not all at once. Yeah.
So you mentioned a possible way to manage BD is to increase the temperature of ponds. Has there been any studies on like the side effects of that on other like physiological measures or like, just sort of species responses?
Yeah, great. So for those on Zoom, the question was, I mentioned management of BD in amphibian populations or communities using habitat manipulations to change the thermal environment, and whether there's been any work on side effects. Very limited work. This is something that really is just being thrown about. There is some work out of, I think, Australia showing that it's negatively affecting not necessarily species physiology, but habitat requirements. Wildlife just really do think they do stuff different than we think they do. And so there was a species that was talked about as not using trees. And so people were like, this is perfect. We'll cut down all the trees. And then it turned out that they kind of like trees. And that's classic. And yeah, so I think any management like that is going to be a bit rife with consequences and side effects. And I think that's where the landscape component comes in. If we can think about these things from a landscape perspective and potentially manage some sites within a landscape to be, hotter, lower BE sites, that might bolster, the broader community in terms of recovering from, BD outbreaks and die-offs. But answering those questions is something I'd love to do, but that's like a career level question. Yeah. Good one. Nancy.
Also wondering a little bit about kind of wetland restoration and like when you're creating wetlands. Maybe
Yeah.
in areas where they've been lost, and that correlation between temperature and temperature variability and your hypothesis of kind of optimizing both. I was wondering, is that like basic thermodynamics that they're correlated, or is it because warmer ponds tend to be warm because they're small and shallow? Or, I mean, is there something you can do in terms of the bathymetry or something to kind of maximize that relationship?
Yeah. So for those on Zoom, thank you, Nancy, this is a good one. For those on Zoom, the question was thinking about pond thermal characteristics. What drives the covariation between heat and variability? And are there any like physical factors of ponds that we can try to manipulate to change those?
You said it much better than I did.
I've thought about it. I've thought about it a bit more. So I think that you would find a similar pattern in other like pond temperate systems, where a lot of the variability is being driven by these heat waves and by diurnal shifts. And so you end up with this situation where they're coupled. I think in systems where variability, like in the tropics or in stream or any floatic system, I think that this covariation would fall apart a little, which could be a cool opportunity to test some of these concepts. We had data on like depth, width, length, so on and so forth, and fit pretty simple models, but fit models to try to understand if these metrics or like a bunch of other ones were related to those, and they weren't. I think just anecdotally, it's a complex combination of size of macrophytes and surface vegetation, of water clarity is a big one, so tannin staining. Because in my experience, the hottest ponds often are those that are clear enough and shallow enough and with low enough like vegetative growth for sunlight to hit the bottom and then re-radiate. So it's none of that's satisfying. Yeah. More work is needed.
More to do.
Yeah, just like totally gonna punt that one. Yeah. Yeah, Tom.
Yeah, I was wondering if you could talk a little more about the sort of how you explain the striking difference between thinking about the sagebrush birds and the effects on prevalence versus occurrence. Why you're not saying we're seeing a huge effect in prevalence but not occurrence? Is that just like a microrefugia thing, or is that just like yeah.
Yeah. So for those on Zoom, Tom's question was like, explain a little more about sagebrush species, bird species especially, and the differential effect of annual grasses on prevalence versus occurrence. So I think about it not necessarily from a refugia standpoint, but just from like a population of marbles perspective kind of thing. That's weird. It made sense in my head. But from a population biology perspective, where we have this change that's occurring to habitats, it's kind of slow and insidious, this invasion. It's not often coming in this data set, at least, coming along with these punctuated disturbances like fire. And so I think in the face of that invasion, you have marginally less and less nesting habitat, foraging resources. And so I think that the habitat is basically just being degraded slowly and able to support fewer individuals, right? And so within that 1 kilometer square, you may still have individuals. The species is still occurring there, it's still occupied. And so psi is capturing this stable relationship. But the number of individuals within that, and thus the species prevalence, because these species have fairly territorial home ranges, fairly well-defined home ranges, you're just having fewer and fewer home ranges occupied, right? And so if enough of those home ranges within a 1 by 1 kilometer square blink out, over a large enough area, then you would see those effects manifest in the psi component, the occurrence component of the model. Does that make sense? That's how I'm thinking about it. There probably are alternative explanations, and if you have one, you should tell me because I'm like writing this now. Yeah, Nolan,
Great talk.
thank you.
So another pattern about the birds that I've been sort of turning over in my head is the you have to have greater diversity with more invasions.
Yeah,
And got me thinking about sort of top down versus bottom up effects,
Mhm.
whether these invasions by grasses or primary producers more generally behave differently than invasions by maybe a predator of some kind or parasite for that matter.
yeah. Yeah, so Nolan's question was about the increase in diversity in bird communities as annual grass invasion has progressed. And if you would expect to see that in cases of a consumer invasion. Oh, such a good question. There's a lot to work with there. I'll be brief. I think this pattern of increased diversity in sort of intermediate stages of invasion for cheatgrass has been quantified. You can think about it from a diversity of resources standpoint. At least in the middling stages of this transformation, you're adding some alternative patches to the landscape, and so you might, be kind of aggregating these novel, higher diversity communities. And intact sagebrush bird communities are pretty species poor, anyways, and so to my mind, in the absence of like burning them or grinding them to the ground, it's not that hard to add species, at least temporarily, to these communities. So I don't expect that increase is necessarily persistent. And then the second question about the effect of invasive bottom-up invasions versus top-down invasions. It's a really good comps question. I don't know. It probably really depends on what sort of releases happen in the face of a new predator. I'm struggling to come up with any good examples of increased diversity. But you might be onto something. I wonder if that's more general, that bottom up or top down invasions would tend to more unequivocally decrease species richness. I don't know. I hadn't thought about it. Thanks for the question, though. I think that's probably time.
All right, let's thank Richard.